Hi, in this video we're going to talk about clustering your single-cell RNA-seq data based on gene expression. So I would assume that you already have your data processed with, for example, cell ranger block or any other block, and also you perform dimensionality reduction as well as batch correction if needed for your data. Now, to cluster your data, we have a block which is called Leiden Clustering, which uses Leiden algorithm to cluster your cells by gene expression. And here we only have essentially two settings. The first one is obviously your data set that you have to select. And the second one is resolution. Now, for most cases, and maybe if you want to give it a try at first, I would recommend leaving it the default value, which is 0.5. Now, the logic behind this value is the lower it is, the bigger clusters you get. So you get less clusters and they're larger if you set the smaller number, for example, like 0.1. Or if you want to have more clusters and they will be smaller, then you might want to increase this number up to say 1.2, 1.1, 1.2. When do you want to do that? So you might want to change it if you know the biology behind your data. For example, if you know that you have a single cell line, a single cell population with clusters that will be hard to distinguish based on the gene expression, you might want to increase the resolution. But if you know that your data has a lot of different cell types and you just want to distinguish between them, the default value is probably going to work fine. So I'm going to click run here, and in a second we should get the result. So yeah, right away you can see here we get, let me close the settings, we get a UMAP with our clusters. These are cluster IDs, and these are clusters that you can see on the UMAP, and as well you can also look at the t-SNE plot and check how these clusters look at the t-SNE plot. Now, these clusters are ready for downstream analysis, and in the next videos I'm going to show you how to work and what you can do having this clustered data. Thank you.